Great. Thanks, Mark and Camille. Yeah, I'll just go ahead and introduce our presenter today, Carolyn Boudreau. Carolyn is a fourth-year graduate student in Harvard's PhD program in virology, where she works in the lab of Dr. Galit Alter at the Reagan Institute of MGH, MIT, and Harvard. Her research focuses on the immune response to the flu vaccine using high-throughput serology experiments and computational systems biology analyses to develop an understanding of how vaccines can be improved. Prior to her PhD, Carolyn graduated with a Bachelor of Science from Tufts University and worked as a research technician at Massachusetts General Hospital. When not in the lab or bike commuting across the city, she can be found reading books, baking classic recipes, and kayaking the rivers and lakes of Greater Boston and Cape Cod. And now I'll turn it over to Carolyn for her talk, Understanding the Novel Coronavirus to Make a Vaccine.
Thank you, Lisa. Let me just share my screen with you all. Okay, let me just clear this out. Great. All right, so thanks again, Lisa, and I want to thank all of the organizers, Lisa, Mark, and Camille, as well as all the other organizers with the Harvard Graduate Students Union for putting together this series and also for all the work that they do to support us graduate students across Harvard, and I am proud to be a member of the union. And thank you to everyone who's listening right now for joining us tonight. I'm happy to have you here. So, a brief disclaimer before I start my talk. As I'm fond of saying to people who ask me for medical advice, I am not and never will be that kind of doctor. Nothing in this presentation is meant as medical advice. And so if you or someone that you care for has symptoms of COVID-19, I would encourage you to please contact your physician, or a lot of hospitals have set up nurse call lines where you can speak to a nurse and get advice about your specific situation. That being said, even when a vaccine or treatment becomes available, please continue to follow the guidance of the CDC and your local government on things like masks and physical distancing. Those are very important to stopping the spread of disease. So tonight's talk is really to help all of you understand and evaluate all of the news that's coming out about vaccines, and to introduce you to the basics about SARS-CoV-2 and how vaccines are made to make you more informed consumers of all of this news. So as Mark said, like the previous talks in this series, I will be stopping at the end of each of my sections to answer any clarifying questions, but I'll save longer, more discussion questions for the end of the talk. And so if you put those questions into that Q&A panel, all of your fellow attendees can see it and I can see them as well. So my talk tonight will include three main sections. First, I'll introduce the virus, SARS-CoV-2. Then I will talk about current vaccine strategies that are being used for SARS-CoV-2. And some of the pros and cons of those. And finally, I'll talk about how vaccines are made and tested and what are some specific challenges for the SARS-CoV-2 vaccine. So starting with that introduction to the novel coronavirus, SARS-CoV-2. So SARS-CoV-2 is the virus and COVID-19 is the disease that this virus causes. SARS-CoV-2 was rapidly identified as the source of the outbreak in China. So in mid-December in Wuhan, China, doctors reported pneumonia with no known cause. And this actually triggered some surveillance and contact tracing protocols that were created after the initial SARS epidemic in the early 2000s. Eventually, China did notify the World Health Organization per international protocols, and the Chinese CDC shared the genome sequence that they had discovered of this virus about a month after the first known cases at that time. So this virus was identified using two different technologies that have really developed a lot in the past few decades. So the first is electron microscopy, and that's what you can see in this image. This is a colorized electron microscope of SARS-CoV-2. And so the reason that the coronavirus got its name is actually really evident in these pictures. Similar pictures to this were taken of different coronaviruses, and scientists looked at them and said, wow, these little spikes on the outside here, they really look like a crown. And so corona means crown, and that's how the virus got its name. So these electron micrographs told scientists that this was a coronavirus that they were working with, and then genome sequencing told them more about what specific type of coronavirus this is and what viruses it's closely related to, which allows us to start work on vaccines and therapeutic strategies. So SARS-CoV-2 is a beta coronavirus. And while that might not mean a lot to most of the people in this audience, to a virologist, it tells us a lot about this virus because we know a lot about how beta coronaviruses behave. There are now 5 known human beta coronaviruses. So there's the newly identified SARS-CoV-2. There's also the original SARS virus, as well as the virus that causes Middle Eastern respiratory syndrome. There are also 2 coronaviruses, beta coronaviruses, that cause winter colds in humans. So most people listening to this talk have probably had a coronavirus before. It's just been one of these 2 milder ones that gives you some sniffles, maybe a sore throat, but you get better on your own. Another thing that knowing about this virus tells us is what the virus life cycle is. So really briefly, to walk you all through this virus life cycle, the virus enters a cell by binding to a cellular receptor with one of its proteins, and then fusing its membrane and depositing its genomic material, or RNA, into the cell. This viral RNA is then translated by the cellular machinery to create the viral polymerase protein, which can then replicate this RNA. This escalates and produces all of the proteins needed for the virus. When all of these proteins and the RNA are created inside of a cell, the virus self-assembles and then buds out of the cell membrane to go on and infect more cells. So just by knowing that SARS-CoV-2 is a beta coronavirus, we can know that it likely follows this same viral life cycle, and this can tell us a lot about how to target a vaccine or a therapeutic. So let's zoom in on this virus itself. SARS-CoV-2 is an enveloped virus. This means that it has a lipid membrane around it, and this lipid membrane, much like the membrane of your cells, kind of holds the inside on the inside and keeps the outside on the outside. So on the outside, embedded into this membrane, are a few proteins that help the structure, called envelope protein and membrane protein. And this S protein shown here in red, or spike protein, which sticks out and is the protein that the virus uses to bind to the cells. On the inside of the membrane, there's the RNA, or the genome, the genetic material of this virus, and that's wrapped around nucleocapsid protein, kind of like thread around a spool. So focusing in on this S protein that sticks out of the virus, the protein structure was solved very rapidly in less than 2 months. If anyone on this call has done any crystallography, they know that this is usually a years-long endeavor that was done very rapidly following the identification of this virus. And so what's shown in the bottom here is a space-filling model along with a ribbon model of the shape of this S protein. And so this tells us a lot about how this protein can interact with proteins on a cell, and it also tells us about how we can target other things to disrupt this protein, because they're going to have to interact with these surfaces. So protein structures allow us to understand how viruses enter cells, how they bind to our cellular proteins, how the immune system sees the virus, because this is the protein on the outside that our immune systems are going to recognize. And like I said, how we can target drugs and vaccines. This S protein, in addition to being on the outside of the virus, is also likely to be expressed on the outside of many infected cells, which means that this is a way that our bodies can recognize those cells that are infected with the virus and clear them before they go on to create more viruses. So the immune system, like us, targets SARS-CoV-2 S protein specifically. So a little bit more about this S protein is that this is the protein that binds to the ACE2 receptor on human cells. So ACE2 is a protein on the cells of your lungs, your arteries, your heart, your kidney, and your intestines. And ACE2 usually works to help regulate your blood pressure. So it's a really important protein. And the virus just hijacks it to enter the cell by binding to it and then changing shape and folding so that it pinches the viral membrane and the cell membrane together. This mediates viral entry and the release of the RNA genome into your cell, where it can go on to replicate and make more viruses. The S protein is also the main target of neutralizing antibodies, which I'll talk about in a few slides. So how does immunity to viruses and to SARS-CoV-2 specifically develop? So I'll walk you through some of the quick basics so you can understand what I'm talking about later on. So viruses inside of your body get eaten up by antigen-presenting cells, cells like dendritic cells. And these are cells of the immune system that are really good at not getting infected by most viruses. And so when they take in this virus, they can digest it into little chunks and then display it on the outside to other cells of the immune system, kind of like saying, hey, this thing is the bad thing. You should recognize it and get rid of it. So these cells include T helper cells, which, like their name suggests, go on to help other cells boost the immune response. And these other cells include B cells, which make antibodies, which I'll talk about on the next slide, and cytotoxic or killer T cells that can recognize infected cells of your body and get rid of them before they have a chance to produce more viruses. So when we're making vaccines, antibodies are really a thing that we want to focus on a lot. An antibody is a Y-shaped protein that looks like this, or a diagram of it looks like this. And it has two main parts. The first part is shown in gray here, and the tips of this gray part are what actually binds to a viral protein. So this is the part that recognizes the virus and knows that it is not part of your body. And then this part shown here in red signals to the rest of the immune system to help it boost immune response against that virus. So antibodies recognize the virus both inside the body and in mucosal sites, like in your nose and your respiratory tract. And antibodies play multiple roles. They can block viruses from entering cells by harming their proteins, by getting in the way of their proteins and preventing their function. They can also act as flags to mark a virus as something foreign that your body should get rid of. And they can recruit other cells of the immune system to a site of infection to help them clear the infection. And so in this way, SARS-CoV-2 virology is really serving as the backbone for our vaccine and therapeutic development, which I'll talk about in the next section. But I want to make sure that everyone is on the same page for the virology. So if you have any questions, please submit them now to the Q&A. All right. So I'm not seeing any questions, so we're gonna forge ahead, and I do see one question. Okay, so Matt asks, how do B cells make an antibody to fit every virus since they're all different? So this is actually, I think, very interesting. So the genes that code for antibodies inside of your body actually are kind of unstable. They can be really easily mutated. So first of all, there's lots of them, and everybody has slightly different ones. And then second of all, they can be easily mutated. And so your body produces lots of antibodies. It checks to make sure that each antibody isn't going to react against any of your own proteins. And then once it knows that, it puts it out, and just a little bit, and it probably won't bind to anything very well. But every once in a while, one of these randomly produced antibodies binds to a virus really well. And just by like a sheer numbers game, our bodies are able to find these antibodies that bind kind of well and then mutate them further and further and further by testing their fit against the protein of the virus in order to create a better binding antibody. And so in that way, we're actually capable of developing this response. But as you might have seen in the testing news recently, this does take time. So people who are COVID positive by the nasal swab test might not be antibody positive for up to a week or two later. All right. So moving on to the second section of the talk. We currently have 123 vaccines as of 2 days ago, which was the last time I checked, that are being developed around the world against SARS-CoV-2. Now, I imagine that most of you don't want to stay here all night, so I'm not going to go through all 123 of them, but I will go through some of the broad categories so that you can evaluate news about these vaccines. So nearly all of the vaccine candidates currently in testing are targeting the S protein, this spike protein that I've been talking about. And some of them go even more specific to target just a piece of this S protein called the receptor binding domain. And as that name suggests, this receptor binding domain, shown in blue in this ribbon diagram, is the part that actually interacts with the ACE2 receptor on human cells that allows the virus to enter the cell. And so this receptor binding domain, if you can block it, is actually able to block infection of new cells by an antibody. So you can imagine that if there was an antibody that came in and bound right to this blue piece right here, the virus would never be able to bind to your cell and enter and create a productive infection. So that's the reasoning behind that targeting, but a lot of them are using the full S protein as well. So now I'll go through some vaccine technologies that are being used, because lots of new technologies are being used in the race to develop a SARS-CoV-2 vaccine. The first to go into trials was an mRNA vaccine from Moderna here in Cambridge, Massachusetts. So this vaccine uses genetic material, specifically RNA, that codes for SARS-CoV-2 spike protein inside of a lipid particle. So it has a membrane, kind of like a cell or a virus has a membrane, and inside of it is genetic material that codes for the production of the S protein. And so this is injected into an intramuscular injection, same as like a flu shot or a standard vaccine. And it causes your body to actually produce the S protein. So this doesn't infect you, it doesn't make you sick, it's just a protein. But it can teach your immune system that this is something foreign that it should recognize. So this viral S protein that's produced by this vaccine trains the immune system without producing any infectious material. It also doesn't require the production of any infectious material to make the vaccine, so it's easier for the people to make it to avoid getting sick as well. This technology has been tested in the past, but is not yet in use for any vaccines. It's relatively easy to produce mRNA, so that's a positive for the development of this vaccine quickly in order to treat everyone during a pandemic. But because it's not yet in use, we're not sure that it's going to be fully safe, and that's why it needs to be fully tested before it's used. The next vaccine technology we'll talk about tonight is DNA. So like the mRNA vaccine, this is injecting genetic material into you to produce the S protein, which will then train your immune system without producing an infection. So again, this does not require the production of any infectious material, but the way that this vaccine is delivered is a little bit different. First, you get a subdermal or just under the skin injection of DNA into your arm usually, and then a special machine that gives tiny electric pulses through microneedles is used to allow that DNA to enter your cells. So this is really easy to produce the DNA. That's something scientists are really good at, and it's not very expensive, and DNA is super stable, so it's easy to ship all over the world. But a definite con of this vaccine is that it requires this special apparatus to administer, and it may not generate that strong of an immune response. And again, this hasn't been used in a vaccine before, so we're not sure how well it will work on a large scale. The next vaccine technology that I'll talk about tonight is the viral vector. So this one might seem a little counterintuitive at first. So viral vectors use actually a different virus. And so instead of the virus hijacking our cells to produce themselves, we're going to hijack the virus to produce immunity. So some of these use adenovirus or vaccinia virus, but essentially what they do is they take out most of the genetic material of that virus, rendering it unable to cause its normal infection. And they insert the gene from the virus you want to create immunity to. So in this case, it's that SARS-CoV-2 S protein. And so when this virus infects your cells, instead of causing an illness, it causes your cells to produce the S protein. And so in this way, it will train your immune system to respond. This can be given by injection or inhalation, and it generates really strong immune responses and is likely to only need one dose, which is great. But unfortunately, it's again not yet in widespread use, and there may be problems with people who are already immune to the virus used as the vector, depending on which one is chosen. Going into a more traditional vaccine approach, recombinant protein approach. Basically for this one, instead of having your body produce the S protein, we're producing it in the lab and then injecting that into you. So they produce SARS-CoV-2 S protein in cells in a lab, And it can be modified to target the immune system in different ways, or to become more stable. And then this is injected into the body. Again, it does not require production of any infectious material, and production facilities already exist for this, but it's not always that immunogenic. It doesn't always create that great of an immune response. So it may need an adjuvant or a type of a booster to get a better response, and it may require multiple doses. And we won't know how that works until we're able to test it. The next vaccine technology is virus-like particles. So these are really interesting. It's like the outside of the virus with no insides. So they're actually produced in plants, and they contain the proteins from the virus, but no genetic material. So it's like injecting an empty virus into you. It can't replicate, it can't make you sick, but it gets all those proteins into your system to train your immune system. So again, this is not infectious, but we don't know how well it's going to work because, again, it's never been used before. The final technology I'll talk about tonight is the inactivated virus. So this is like the real traditional kind of vaccine that's used. This is what's in a lot of, for example, flu vaccines. And this is a virus grown in a lab, inactivated with chemicals or radiation, and purified. Then this is injected, and it's typically somewhat immunogenic, but again, because there's a finite amount of the virus going into your system, you might not get that great of an immune response. It is the traditional strategy. We're pretty good at it, and we have existing infrastructure to make it. But unfortunately, it requires us to grow a lot of virus. So it can be dangerous for the people who work there, and they can have to go through a lot of really strict safety procedures. And again, it might not produce the most robust immune response. So that's all the vaccine technologies that I'm going to share with you tonight. So I will stop here and see if there are any specific questions on this section of the talk, and any others I will get to at the end. Of the vaccines that we're familiar with, for example, flu and measles, which vaccine technology did they use? Are some of the methods more common than others? So this was a question from Mel. Yes, so a lot of vaccines currently in use either use inactivated viruses or recombinant proteins. Those are the most two common ones. The flu vaccine, for example, because it happens to be the one that I work on, I know the most about it. The most common one that people get is an inactivated virus that's grown in eggs. If you are allergic to eggs, you likely get one that is grown in cells, and so is a recombinant protein vaccine. So these two are the most common. The others are very experimental, and they've been tried in animal models and some small scale trials, but they haven't been tried at a large scale yet. Let's see. Abby asks, are there any vaccines in development that target T-cell responses? Well, I can't say no because I don't know about all the vaccines, but I don't know of any that are really strongly focused on T-cell responses. I think a lot are hoping to find that ideal balance of interaction between the antibodies and the T-cell responses. But I know that based on information that we know from the original SARS virus, a lot are really focusing on the elicitation of neutralizing or sterilizing antibodies that block the virus from infecting cells. Anonymous asked, since we aren't certain whether humans can develop natural immunity to COVID post-infection, is there uncertainty about whether a vaccine is possible? So the current understanding, as far as I'm aware, is that most humans do develop at least short-term immunity following infection. That was true for the original SARS virus, which is about 80% similar to this virus. And it seems to be true with the reports coming out from areas where the virus hit earlier. There is always uncertainty about whether a vaccine is possible until we've made one. But I think we have a lot of reasons to believe that we could make a vaccine. Unlike things like HIV, where the vaccine effort has been going on for many years, this virus does not seem to be mutating quickly, and so it's not likely to escape a vaccine in the same way that HIV would. So the short answer is yes, there's always uncertainty. That's how science works, but I think we have good reason to believe that it is possible. Anonymous asks, for all of the methods that cause your cells to make the viral protein, does the protein need to come out of the cells before antibodies can be produced against it? That is a great question, and the answer is yes, it does. The S protein will actually be transported by your cells from the inside of the cell to the membrane because it needs to be on the membrane of the virus, and so it has a signal encoded in it that targets it there. And so that will get some of it out of the cells. Other parts will come out just by like overproduction. If there's too much of it in the cell will push some of it out, and also dying cells. Anonymous asks, what determines how immunogenic a vector is? So there's a lot of components that go into this question. It's a great question, and if we understood all of it, we'd be making much better vaccines much faster. A lot of it has to do with the amount of exposure and the duration of exposure. Those are two big things that can affect immunogenicity. So how much of this protein are you putting into the system of a person's body, and how long is it going to stay there for? Is it only going to be there for a day? That's not great. Or is it going to stay there for a couple weeks, maybe some months? That's much better.
Carolyn, I think since we finally have people, the Q&A is finally flowing, I'm wondering, would you want to finish up the last section and then come back to questions, or do you want to take a few more now?
Let me see. If I can take, there's one real quick here. Stu asks what kind of plant produces a virus-like protocol. That one specifically, they're not produced in nature by plants. They're grown in plant cells in a lab, and that one specifically is made using tobacco cells. Okay, I think I will save the rest for the end. Thanks, Lisa.
Thank you.
So for our third section, how vaccines are made and tested. So this is a typical vaccine production pipeline, and it takes generally more than 10 years to produce a vaccine. Looking across this typical vaccine production path, following the academic lab, finding a discovery that might lead to a vaccine candidate, a pharmaceutical company performs preclinical research through a partnership to determine if this vaccine candidate is viable. This might include studies done in small animals or just studies done in cells in the lab. Then there are 3 phases of human testing before it's approved for use in a wider human population. Additionally, while all of this testing is occurring, manufacturing and quality control testing are implemented and scaled up. So I'm going to walk you through each of these steps. So like I said, preclinical testing is critically important, and this is where a lot of vaccine candidates sink or swim, so to speak. So one of the really important things during preclinical testing is quality control. The final product of a vaccine needs to be high quality, consistent, and safely produced for use in humans. This means that they have to follow really strict manufacturing protocols, ensure all the regulatory requirements are being met, and this can take a lot of time and energy. So one vaccine company that I read about estimates that 70% of the time in vaccine production is spent only on quality control. So this makes sure that the vaccine that's being delivered to all of us is clean and safe and hopefully won't cause any side effects due to the manufacturing. The second thing that's tested is safety in animal models. We really want to be sure before we put this vaccine into humans that it's not going to hurt anyone. And so we look for unintended side effects in animal models. Animal models aren't perfect, but they can tell us a lot. Once preclinical testing is finished, vaccine testing in the United States, as well as most of the world, goes through 3 phases typically. So phase 1 is very small, it's less than 100 people, and it's really looking to see, is this vaccine safe? It may also look at some immune markers of how well the vaccine is working, but it's really focusing on, is it safe? Are there any serious side effects? And how much do we need to give? What is the dosing? Phase 2 includes several hundred volunteers. It looks for most common side effects, things like redness and swelling at the injection site, headaches, things like that, smaller, more manageable side effects, as well as, again, how are the immune systems responding to the vaccine? And phase 3 trials involve hundreds or thousands of volunteers. And this is where we really get a sense of if it's working, because in this case, presumably some of these people would be exposed to the virus. So we want to know how these people who get the vaccine and how people who do not compare. Do they get sick at the same rates? Do they get well at the same rates? Is the vaccine safe and effective? And what are the most common side effects so that we can warn people going forward? So vaccines have a really high bar for safety and low side effects because they're given to healthy people. So unlike a drug that's given to somebody who's already sick, and maybe you can handle some more severe side effects if it's going to make you well eventually, vaccines are given to people who are in good health. And so it's really important that they don't cause any severe side effects. So here's the timeline that I showed at the beginning of this section, and now with this bottom section for updates as to how vaccine production might occur during the current pandemic. Targets have already been identified using previous research on other coronaviruses and other viruses in general, and then clinical development will be accelerated, in some cases by merging the phases of human trials together or doing them very rapidly in succession. And through this, these vaccines can be tested more rapidly, and we might be able to make some choices that we wouldn't make outside of a pandemic situation for the good of all of us as a whole. And unfortunately, I think what is going to be one of the big issues going forward is the manufacturing of these vaccines, because that can only be accelerated so much, and we're limited by the current global capacity. So just to give you a quick sense of where some current vaccine candidates are, this is by no means a comprehensive list, but here's a few things. There are a lot of vaccines in preclinical trials. There are a few that have moved into phase 1 trials in the United States and Europe, as well as in China. And there are a few in China that are in combined phase 1, phase 2 trials. So now I'm going to talk about some of the challenges to SARS-CoV-2 vaccine development and why it's not going even faster. So the first one is that we're not sure what the correlates of immunity are. So what I mean when I say correlates of immunity are markers that can tell us who's immune to the virus without testing by trying to infect them. So this we think is going to be neutralizing antibodies, but we're not sure yet. Or it could be something that's just a marker that doesn't actually cause you to be immune, but develops alongside immunity so that we can test who's immune and who's not without infecting them. So this allows us to really test vaccines much more easily if we can take a blood draw and say, how many neutralizing antibodies do you have, instead of waiting and seeing if you get infected. So correlates can be discovered in multiple ways. We can get a preliminary idea of what they might be from animal models, but of course animals aren't people, and so they don't always translate one-to-one. Another way that we can discover correlates of immunity is through convalescent individuals or people who got sick and got better. We can look at what's different between people who got sick and got better and people who got sick and unfortunately succumbed to the disease, and try to figure out what caused those people to get better that the other people didn't have in their immune systems. And finally, in large-scale vaccine trials, we can see what's different between people who got the vaccine and were protected versus people who maybe didn't get the vaccine and weren't protected. Another challenge to vaccine development is antigen design. As I alluded to earlier, there are currently vaccines targeting both the full-length S protein and the receptor binding domain, based in part on information we had from the first SARS outbreak and also from new research coming out in the last few months. In the last few days since I made this slide, there has been a lot of research into the N protein and how many antibodies might be present towards that. And so there are some scientists who are saying that perhaps we should also be targeting that protein, but that remains to be seen. Another challenge to this vaccine development, particularly in the preclinical stage, is the lack of good animal models. So mice are the most commonly used animal model in vaccine research, but differences in the ACE2 gene in mice actually protect them from being infected. They don't really get sick with SARS-CoV-2. And so one way to work around this is to produce genetically modified mice. And so that's being worked on so that they express the human ACE2 receptor instead of the mouse one. But that will take some time to create. Likewise, rats are also protected by differences in their ACE2 gene. However, hamsters actually have similar transmission dynamics to humans of SARS-CoV-2. If you get one sick, it might cough on another one, and then that one will get sick. And they do get SARS-CoV-2 in the lungs, so it might be a good model to use moving forward. Ferrets, which are commonly used for other respiratory diseases, do not have the same symptoms from SARS-CoV-2 as humans. And non-human primates or monkeys do seem to get asymptomatic infections like healthy young humans do, but we don't know yet how monkeys with some of the same risk factors as we know about in humans might respond to the virus. A final challenge that I'll mention is just that the SARS-CoV-2 vaccine has to be very careful that we don't end up with disease enhancement. So this is kind of rare. It only happens that we know of for a few viruses, but we do want to be very careful because it's been observed with SARS in animal models. So there's two different ways the vaccine might make the disease worse. First, there's antibody-dependent enhancement. So this is what's been observed in SARS animal models. And basically, it's when the antibodies actually, instead of protecting you, help the virus get into your immune cells. So the virus is bound to the antibody is bound to the immune cell, and instead of triggering an immune response, it triggers an infection. So this happens in dengue virus. We're not sure that it happens anywhere else, or at least I'm not sure. I haven't seen the evidence that it happens anywhere else, but it is something we really want to be aware of. The other thing that can happen with antibodies is you can just get big immune complexes. So this is viruses and antibodies all forming like a big clump. And this clump can cause a problem because it just activates every single immune cell in the vicinity. And then those immune cells can like get a little too excited and start damaging your lungs or other tissues. There's also cell-based enhancement of disease, which was seen in one vaccine trial for the original SARS virus, which occurred via allergic inflammation in T cells. So again, this is just like an overactivation of the immune system that we want to be careful to provoke the type of immunity, the specific flavor that's going to protect us and not the flavor that might make us sicker. But this is all what all that testing is for. So I hope that I have shown you tonight a little bit about the virus SARS-CoV-2, some current vaccine strategies, and how these vaccines are being made and tested. And I will take all of your questions. Before I do that, I just want to leave you with two big thoughts to think about going forward. These are things that I don't think anyone has answers to yet. So vaccine development is happening faster than ever, but is it going to be fast enough? So the Ebola vaccine following the 2014 outbreak took about 5 years to develop, but now does exist. Zika vaccine research was on track to possibly be record-breaking and produce a vaccine in 2 years, but when the epidemic died down, so did the funding, and so those labs and companies were unable to finish testing that vaccine. So in this graphic on the bottom left here from this really great New York Times Interactive shows the typical vaccine production timeline based on their research wouldn't get us a vaccine until the mid-2030s. And yet we're trying to accelerate this all the way down to mid-2021. And so just how quickly all of those things need to happen. And why do we need to do it so quickly? Well, that they illustrated with this graph on the right, which shows the deaths over the AIDS epidemic from the 1980s to the late 2010s, and how many deaths there were per year, and that we've already exceeded that, and we're not even halfway through the year yet with our deaths from COVID-19 this year. And once a vaccine exists, who do we give it to? So this is a moral and ethical question that I'm not going to even try to answer. But will we give it to healthcare workers and first responders? Will we give it to high-risk people? Will we try to contact trace and give it to anyone who's been exposed? Will we give it to the people who can pay for it? That's something I think we as a society are going to have to answer. And so I just wanted to put it out there for everyone. So with that, I would really love to thank you all for joining us. I did leave plenty of time for questions, and I've posted some of my favorite resources here if you are interested in learning more about vaccine development.
Thank you, Carolyn. Are you going to be sharing the slides with everyone after?
Yes, I can share the slides afterwards.
Okay, great. Everyone will be able to check out those links later. And yeah, so the Q&A is still open. Right now we have 13 questions. Carolyn, do you want to go ahead? And yeah, that was a great presentation too. I think I did not say that.
All right, I'm just going to try to go through these questions mostly in order. So Amos asks, In what ways does a virus like SARS-CoV-2 mutate, and how does that affect the efficacy of potential vaccines? Are some potential vaccines better at dealing with mutations than others? So far what we know about SARS-CoV-2 is that it does not seem to be mutating very quickly. So unlike the flu or even more severely HIV, the proteins that the immune system targets on SARS-CoV-2 don't seem to be changing as it spreads around the world. And so this actually boosts the efficacy of all the potential vaccine candidates because it means that the proteins you're exposed to in the vaccine are more likely to look like the proteins on the virus. Are some potential vaccines better at dealing with mutations than others? I don't think that any of the vaccine candidates that I described here would be better at dealing with mutations. However, some of them would be easier to change if the virus mutates. So those that involve genetic material, so the DNA vaccines or the RNA vaccines, would be very easy to switch production to a new sequence if the virus changed. But again, we would then have to revaccinate everyone. So it's still not great, but it would be easier to produce than some of the others. All right, Anonymous asks, what determines how long vaccine particles stay in the body? And does this vary by the main vaccine mechanisms that you described? So that's a really great question. So vaccine particles in the body, it depends which kind of vaccine it is, but I will talk about the ones we know more about, the more traditional vaccines, so the recombinant protein and the inactivated virus. So the length of time that that stays in the body is really determined by how it is injected and what it is injected with. So the way that you formulate the vaccine, not just what the protein is in the vaccine, is very important to that. I would say it's on the order of weeks, typically for most vaccines, but that's not always true. And yes, this does vary by the main vaccine mechanism, absolutely. All right, a lot of anonymous questions. I don't know if this is all the same anonymous or this is many anonymouses. Is the fact that SARS-CoV-2 targets S proteins part of why it's so virulent? So SARS-CoV-2 doesn't target the S protein. SARS-CoV-2 has an S protein. So the S protein is the spike protein that's found on the virus, and that targets the ACE2 receptor. So on human cells, that ACE2 receptor is also targeted by SARS, the virus that caused the epidemic in the early 2000s in Asia. Those are the only two that I know of that target that protein, but I can't say definitively that there aren't others. Norm asks, how can the body differentiate between the natural binding partner of ACE2 and the S protein? Aren't they shaped basically the same? So that's a really great question. No, they're not necessarily shaped the same, sorry. So the S protein and ACE2's natural binding partner are likely shaped very similarly at that specific receptor binding domain, but again, not exactly the same. And the rest of the protein might be shaped very differently. And so an antibody targeting that might hit some portions that look similar to a human binding partner, but some portions that look different. Anonymous asks, if another country develops a vaccine, what is the FDA approval process? It depends what country it is. It will need to be tested to some extent in the United States, but what country it is determines how much testing will be required to be done. And I don't know the details for every country, sorry. Let's see. Surya asks, do we have manufacturing set up to scale up all the kinds of vaccines if approved, especially given that mRNA vaccines have not yet been tested? That is something I've been thinking about a lot, and the answer is no. We don't have manufacturing set up, and not just we being the United States, we being the world, don't have manufacturing set up to produce billions of doses of any of these vaccines. Personally, my opinion is that the best case scenario would be if multiple of these different types of vaccines work so that we can be producing different ones at the same time so more people can get vaccinated. Let's see. Another anonymous, is it possible that different individuals produce antibodies that recognize different parts of the virus? Yes, absolutely, definitely happens. So most people have lots of antibodies that recognize all different proteins and all different parts of different proteins, but the ratios of those antibodies in different people can vary a lot. Bethany asks, I know the flu vaccine needs to be given every year because the flu virus mutates, leading to a lapse in immunity. Do we know how long a vaccine might last once we have one? And is there anything that can increase the length of efficacy? Yeah, so as I mentioned before, it doesn't seem like this virus is mutating much. So we don't have a strong thought that we'll need to give more doses for that reason. However, immunity to different viruses lasts a different amount of times. For example, you probably got vaccinated for measles when you were very young, and you never need to be vaccinated for measles again. Most people will have that immunity their whole lives. On the other hand, there's things like tetanus, where you need to get a booster every once in a while, or things where we don't have a vaccine, like norovirus, where you might get infected and you're immune for a little while, but a few years later, you're probably susceptible again. And so we don't know how long a vaccine would last in terms of creating immunity. From the original SARS epidemic, it looked like the survivors had immunity from infection that lasted about a year or two, but we really don't know with this virus. That's just a guess. So let's see. Alina says, can you tell us a bit about how the CDC and corresponding entities in other countries have modified or relaxed the approval processes or guidelines for vaccine development in response to the urgency of the need? And what are my thoughts on the trade-offs? So I will start by saying I'm definitely not an expert in FDA approval processes or CDC testing processes at all. I think that a lot of what they've done is lowered the bar for going into initial stage testing, where they're testing in maybe 20 or 30 people to make sure that something is safe, as long as they have some level of animal data, not nearly as much as they would normally be required to produce. The other thing that I know that some different agencies around the world are doing is letting people start phase 1 trials, and then just a few weeks to months later, as soon as they know that those people aren't having any serious response, they're going straight into a phase 2 trial. And so they're accelerating the time between those trials, as well as getting into the trials. I think a lot of these are wise decisions given the urgency of creating a vaccine right now. I totally understand why they don't usually do it this way, and I don't think we should always do it this way. But I do think because all of us really need to get back to living our lives the way we're used to, we are going to have to make some trade-offs there. But it is difficult, and I don't envy their job. Let's see. Anonymous asks, if it takes some time for people to develop immunity from getting a vaccine, how would ringing work if those potentially getting the vaccine in that model have already been exposed? That's another great question that we're definitely going to have to take into account. A lot of previous ringing strategies went not just to that first ring, but the ring from those people that those people are exposed to, take it that one step further or even 2 steps further. But again, those were not things that were happening all over the world all at once. That's an unlikely strategy to be used if the pandemic is still looking the way it is now. That's a more likely strategy to be used if it's just a case pops up here, a case pops up there, if it goes down to that level. Let's see. Matt asks, I've heard about the possibility of a challenge trial being used to speed development and testing time, where healthy volunteers would be deliberately infected with the real virus after administering the vaccine in order to test the efficacy of immunity. What are your thoughts on this, and is there an ethical way to do this? I'm not an ethical expert. Typically, that kind of trial is not done for things where we don't have a treatment. So it is done regularly for things like malaria, for example, where we don't have a vaccine. We really like one, but we do have treatments that can cure people. So people are actually given vaccines and then intentionally bitten by malaria-infected mosquitoes. And then if they're monitored very closely and if they get sick, they're given treatment. Because we don't currently have a treatment for the COVID-19 and we can't even fully predict who's going to get really ill or not, I don't know if there's an ethical way to do this right now. I think if we were to have a treatment, if some of the drug trials that are underway right now have positive results, I definitely think that that's a very ethical way we could create a vaccine. But right now, I'm not sure. Barry asks, there are vaccines for coronaviruses in cattle. I didn't know that. What type of vaccines are used for coronaviruses in cattle, and has this information been helpful? I'm sorry, Barry, I didn't know that. You taught me something tonight. I will go look that up. Thank you. Anonymous asks, is the reason why COVID-19 isn't mutating because it's so contagious and easy to spread, or is there another reason? So mutation rate of viruses depends on a lot of things. One of the main things that it depends on is how error prone the viral polymerase is. So when it's making copies of its own genetic material, how many errors is it introducing? So the coronavirus presumably has a polymerase that is not particularly error prone, but I don't think it's because it's contagious. That contagiousness and mutation don't really affect each other in that way. Christine asks, can you explain what we know about the inflammatory response in the lungs that affect some critically ill COVID patients? What is happening that is causing this reaction? I wish I knew, Christine. I think it would help a lot of people. I don't know. I don't know if anyone knows what is exactly happening. One thing that scientists think is likely the cause of this is what's called a cytokine storm. And so that gets towards this inflammatory response that you mentioned. Basically, the immune cells are getting way too excited, and in the process of trying to destroy the virus, they're causing a lot of collateral damage to the body. And so we need to figure out ways that we can boost the type of immune response that's going to get rid of the virus without harming the person. Anonymous asks, do you know if NEEDS at BU is doing COVID work? I'm going to guess you're asking about the needle, NEIDL instead of NEAD. I'm sorry if that's not what you mean. I'm not familiar with NEEDS, but I do know the needle, which is the BSL-4 facility at BU, and yes, they are doing COVID work. Anonymous asks, do you know why there's only one group company working on an inactivated virus vaccine if it's such a common approach? I'm not sure that there's only one. My slides are probably not comprehensive for all the different companies working on this. There were not 120 down there. I do know that it's a less common approach right now for this virus for two reasons. One, we don't think it's going to have a great immune response. We think that it's likely that the body will just kind of clear it out like trash and not really respond to it. So a lot of people are working on things that are more likely to generate a robust immune response. The other reason is that it requires growing giant vats of infectious virus. And right now you have to do that under very strict biosafety conditions. And so it's expensive and time consuming and difficult. And we're unlikely to be able to produce enough of it for everyone. So I think that's another big roadblock in the way of that one. Mohammed asks, some countries are already using plasma therapy from recovered patients. What is my opinion about this? It seems some early reports like maybe it's working. I don't think we know yet. I think it's a great idea. It will be improved if we can tell which recovered patients have the best immune responses. That would be most helpful to those people. So I think we need to learn more about the immune response before that's going to be super effective. I think it's great in emergency situations, but it's not something we can scale up to treat everyone with, obviously, because we're taking it from other humans. Bethany says, Jackson Labs acquired a transgenic mouse expressing human ACE2 from a scientist at the University of Iowa. That mouse is being bred and is now available for pre-order. Thanks for letting me know, Bethany. That's great. That's great news for all the vaccine testing. Anonymous says, is there a difference between antibody tests on the market testing who recovered versus the ones needed to test the effectiveness of a vaccine? So there's two different questions embedded in an antibody test. One is, do you have antibodies? Which is what the test currently on the market will tell you. Some of them, depending on which one, can also tell you how much antibody you have. Ones needed to test the effectiveness of a vaccine would also need to know what type of antibodies are protective. And that we don't know yet. So we can tell you if a vaccine produces antibodies in the virus, but we can't tell you if they're the right kind of antibodies that will keep you from getting infected. Maria has a two-part question. One, would it be possible that this would be a live vaccine or always inactivated, and why? It is certainly possible that there would be a live vaccine. I did not come across a lot of research into that currently during my reading for this talk, but that doesn't mean it's not out there. Live vaccines are typically attenuated in some way, so they're made so that they replicate less. I don't see any reason why this couldn't work. I'm sure someone is looking into it. I would have to do more research to give you a better answer. The second part of Maria's question, I also read something about the protein folding problem related to vaccine development. Is this something that has been discussed and solved? The protein folding problem. I'm sorry, I'm not familiar with that. I don't think I have an answer for you. Sorry, Maria. An anonymous asks, once a vaccine is approved for mass production, is the company that created it the only one that would be able to produce it, or will other companies be able to make it and accelerate the process? That would depend on the companies and the governments involved. I know right now many governments are actually trying to work with companies so that if, say, two different companies are testing vaccines, company A and B, and company A's vaccine work and B's doesn't, that B would also be contracted to produce A's vaccine. I think all the people working at pharmaceutical companies are also people. They all want to help. I think a lot of people would want this to happen. Yes. Kevin, my brother, asks, a preprint study suggested COVID-19 has already mutated. Is this considered a fast or slow mutation considering the amount of infections, and how might this affect current vaccine development? So I have not fully read that study, but I do know that there has been some controversy about how accurate it is, and I think that that is true for all preprint studies. I think that the level of mutation that has been seen so far, at least in things that I have read, has led us to believe that it's a slow mutating virus and that this is not a serious hindrance to vaccine development right now. All right, we're at exactly 1 hour and also through all the questions, but I am happy to stay and answer any more if people have more questions. My email address was on the first slide, and so it will go out if people did want to ask more questions later. Just saw something.
Yeah, so maybe you can give people a couple minutes to see if they have any more questions. But in the meantime, Carolyn, I'm curious, do you have a favorite virus and vaccine you have worked on or would like to work on?
So I currently work on responses to the flu vaccine. So influenza is my favorite virus. I would like to work on vaccine development when I'm done with grad school, but I'm not particular about which virus I would like to work on. Thanks for asking, Lisa. Do you guys know if there's any questions? I can't see the chat, but if there's any questions in there.
No, I think we've been transferred.
Yep, they were all in the Q&A this time.
Perfect. Good job, guys.
Yeah, good job to you, Carolyn.
Yeah, this was wonderful. Thank you.
Yeah, so we'll be hopefully getting the recording out in the next 1 or 2 days. And a little update on night school. Next week, we're taking a break. Then we have 2 more talks lined up the weeks after that. And it sounds like we're going to be doing a capstone discussion panel on June 3rd with all the presenters. So that should be a fun time. And we'll be sending out information to everyone soon about how to watch all of that. So yeah, thank you everyone for joining.
Just a question that came in.
Oh, really? Okay.
I really, either Anonymous is very into this or there's a bunch of Anonymous.
There are a bunch of them, I think.
Do you think a vaccine is necessary for quarantine or social distancing to end? Unfortunately, I don't think a vaccine is going to be ready soon enough. And I think that at some point we're going to have to reduce the level of quarantine that we're in. I think some level of social distancing will be required for a long time and potentially until we have a vaccine. But I'm not a public health official, so please listen to your public health officials.
Yeah, thanks to whoever sent in that question. That's an important one. We have tried very hard to try to get some of our public health, our union members who work on public health, available to speak, but they are all extremely busy with their research, and it's been hard. They've had very tough working conditions the last couple months. That's all the questions, I think, in the Q&A, and people should feel free to email Carolyn. I think that probably wraps it up for tonight.
Thank you everyone for coming and for your great questions. I really enjoyed this.
Okay, thanks so much, Carolyn. Thank you, everyone.